Good afternoon, everyone. Good afternoon. My name is Peyman Nasr. I am an associate professor and chair of clinical science department, and I cannot express how grateful I am to see everyone back on campus. Welcome back to our campus. It is my pleasure today to introduce Dr. Jeffrey Golden, a prominent physician, scientist, and researcher. Dr. Goldman is currently serving as the director of Burns and Allen Research Institute, as well as the vice dean of research and graduate education at Cedars-Sinai. Dr. Golden earned his medical degree from University of Pennsylvania in 1988, followed by an internship in medicine from UCLA and residency and fellowship at Massachusetts General Hospital, and finally a postdoctoral fellowship in genetics at Harvard Medical School. He has an extensive professional career over the past 30 years. To name a few, he has served as the chair of pathology at Brigham and Women's Hospital in Boston. Oh. That's the problem with having mask and glasses. Professor of pathology at Harvard Medical School, president of New England Society of Pathology, and pathologist-in-chief at Children's Hospital of Philadelphia. His scholarly activities are exemplary. It will suffice to say that he has authored 2 books, more than 134 manuscripts in high-impact journals, among which words such as Nature, Cell, JAMA, they are quite commonplace. He has also authored more than 20 book chapters as well as monographs. But my astonishment has been his ability to maintain NIH funding for the last 2 decades. This is something that you see every day, and I should congratulate Dr. Golden for this exemplary record. Dr. Golden is going to tell us about the technological revolution in data analysis and its implication in precision medicine, as well as the opportunities that may rise as a result for students who seek careers in medical fields. So without any further delay, please join me to welcome Dr. Golden to the podium.
Well, thank you, Peyman, and thank you all. It's great to see people in person. I actually have not been to any places where we get to see people in person as well, and so this is really a treat for me as well. What I want to do today is share with you. So let me just get started by saying that what I'm going to be telling you about today is not what my own research has been, but what I've been engaged in for the last little over a decade, which is thinking about how do we really transform how we deliver healthcare by more efficiently and effectively using data. At this time, there is tremendous amounts of data that is captured on all of you and on patients across the country. Yet it's used in an infinitesimal way. And by changing that scenario, we can actually think about changing how healthcare is delivered. But to do that is going to require a new generation of types of individuals working in healthcare that have a sophistication with using data. And that's really what I want to share with you today The first slide you're going to see covers some of the problems we have in healthcare in the United States today. I'm going to show you that the quality of healthcare that we deliver in this country is not at the top of the scale. It's actually way down when you look at all the developed countries. If you look at a measure like maternal mortality, women who die during childbirth, if you look at the top 10 countries, they're all below 10 per 100,000. And in the United States, it's almost double that, nearly 18 per 100,000. Yet, despite things like that, we spend far more per person on healthcare than any of those other countries. But right here, you can see the quality of healthcare in all of these countries, and here's the United States, way down here. Look at maternal mortality rates in New Zealand, Norway, the Netherlands, all the way across Germany, and look at the United States. Double what all of these other ones are or more, triple in some cases. And look at the cost of healthcare in the United States. Here's a very nice linear line. This is all these other countries. I put a few of them in. Norway, Netherlands, this is Germany, United Kingdom, right next to the United Kingdom, that's France. All of these are on a very nice linear line. Ireland actually at a lower cost of healthcare, but the United States way above what you see elsewhere. So what is the solution? And I began to talk about that. There really isn't any single simple solution, and we do need to better utilize data. And I'm going to be talking about that today as we focus on precision medicine. What is precision medicine? These are just a couple definitions. The first one at the top from the National Cancer Institute focused on cancer, and again, I'm going to use an example of that. Down at the bottom, what the FDA defines as precision medicine, which is really focused on not just genetics, because, and I really want to make this point, people talk about the genes, but it's much more than that. It's also the environment. It's their lifestyles. It's about all these other things that happen in an individual. That's what precision medicine is, really to get to an individual. But as we're going to talk about, it's also for populations. We want to really think about how do we better care for populations of patients. And in doing that, we want to make sure patients are getting the right treatment at the right time and in the right location for the right patient. And why is that important? Well, it turns out that many patients start off with the wrong diagnosis, they start getting the wrong treatment, and they have many complications because of that. So again, the sooner we can get the right patients on the right path to therapy, the better off we're going to be. Okay, and I'm going to skip this. So let me just briefly tell you about how much data we're talking about. Now again, I'm a little pathology centric, but I can talk more broadly. But let's focus on pathology for a minute. 60 to 70% of the structured data in the healthcare, the EMR, the electronic medical record, comes from your pathology laboratory. And somewhere around 70% of all clinical decisions are based on one of those pathology results. So whether it's as simple as a patient coming in and they, let's say they need to get chemotherapy, before they can even start their therapy, they're going to get blood counts done. They're going to get some chemistries done to make sure their potassium and magnesiums are okay. Up to complex things like what is the diagnosis. All of those decisions are based on a pathology result. And just to give you an idea of how much data is pathology driven in the electronic medical record, a number of years ago when I was back in Boston, we switched from one electronic medical record to Epic, which is one that many people are using across the country now. And when we did that, the second most common data type that we had to transfer over was radiology. And there were 14 million radiology results we had to do over 5 years. Okay, so pretty big number, right? 14 million results. The same time frame for pathology, 500 million. So that gives you some context, the most common and then the next most common in terms of that type of data. So this is that woman that I was telling you about. And you can see this is her cancer right here. And again, she was 52 years old. She had metastases to the brain. And back in 2004, she would therefore have about a 1% chance of survival for even 18 months. But you can see that she was started on this particular therapy that was targeted against that EGFR mutation, and that cancer basically disappeared. It did start to come back, but we found that she had a second mutation in that same gene, and we were able to start her on a second drug that kept her alive for another 4 years, and she was working during 6 of those 8 years. So it really did change that. And so how does that happen? How do we do that? And I really want to focus on not the specifics of this slide, but the concept of this slide. Because what we developed when I got to the Brigham and Women's Hospital was a panel of genes. It's 462 genes that we sequence. And when we sequence genes nowadays, we put them on these sequencers, and what you get out is data. And that's what the data looks like in this upper left panel. It's all these colored dots. Now, if you gave this to a physician and said, here's the results of that tumor, it's worthless. They cannot do anything with it. So we have to take that data and we have to transform it into information. The information is how those 4 base pairs, the A, G, C, and T's, line up to give us sequences that are in our genome. And so we line that up and we get information. So we've gone from data to information. Now again, if I would attest that if I give this set of sequences to a clinician, they again will not know what to do with it. It has no value to them. So we have to convert that information into knowledge. And I'm going to come back to this path of data to information to knowledge. The knowledge is that this sequence corresponds to specific genes, and if we tell that clinician that, where is it, EGFR should be right in here somewhere, EGFR right here. So, oh, you have a specific mutation in EGFR, and that mutation is going to respond to this particular drug. Now the clinician has something they can do it with. So from a data standpoint, we started at an informatics standpoint, bioinformatics standpoint. We started out with all of this data, we converted it to information, and then we made it knowledge. That's what we have to do across the healthcare spectrum, is we have to do that. And why is that important? Well, what we found is that when we do it, it works, first of all. We find common mutations commonly, but we find unexpected ones, and those unexpected ones allow us to not only discover new types of cancer, but also treat things that otherwise were not able to be treated or being treated in the inappropriate way. So what does this all mean? Well, at least for cancer, what it means is that these specific mutations can be treated, that we have more precise diagnoses, we have better treatment stratification, we get patients on the right treatments from early on, and these therapies are actually more effective, they are less toxic, patients tolerate them better, they have improved outcomes, and believe it or not, despite the fact that these treatments can cost $100,000 a year, they're actually more cost effective. How could that possibly be? Studies have been done, and they've shown that when people go on these targeted therapies, they go to the emergency room less. Every time you go to an emergency room in the United States, it costs you about $10,000. You can imagine a few trips to the emergency room and you've saved $50,000 pretty easily. You have less days in the hospital. An average day in a hospital is $2,000 unless you're in an intensive care unit, then it can be triple or quadruple that. And so you spend, instead of 5 days in the intensive care unit, you don't go at all. That saves another $40,000. pretty soon you're well below the cost of that $100,000 a year treatment. But it's not only that. Because these patients are not going to the hospital, because they're not having to stay in the hospital, they're actually healthier and at work. They remain active in society. And that has another financial value associated with it. And all of these have been looked at and actually measured in different ways. And I just show one study there from a group at the Mayo Clinic. Now, what I've been telling you about is all just related to sequencing, looking at the tumor genome. But in fact, we now know that you can actually take and look at the pathology, the histopathology. So here's, let me just go on in the interest of time here, because we got cut a little short. Here's two different lung cancers. This one happens to be a squamous cell. This is an adenocarcinoma. And as a pathologist, I can look at that and I can tell you, oh yeah, that's adenocarcinoma or that's squamous cell carcinoma. But I really can't tell you what's going to happen with that patient. And so a lot of times what we use is something called tumor stage. So has it metastasized or is it localized? Or we might use the tumor grade. Is it a high-grade tumor, therefore likely to be more aggressive, or is it a low-grade tumor, less aggressive? Now we can take artificial intelligence using convolutional neural networks, so a type of artificial intelligence, have it look at these pathology images, and we can say, oh yes, this is squamous cell carcinoma, and then that algorithm can actually better stratify if that's a patient that's going to progress rapidly or slowly, and therefore change the therapy that we are going to give that patient. So we can do that, and we can even, and I'm not going to go through this in detail, this is a study I did looking at IDH mutations important for brain tumors. We can actually predict which patients with a very high degree of accuracy, 92%, we can tell, even looking at the histology, if that patient has a specific mutation and what's going to happen. That's what the computer can tell us when we look at it. I can never do that with my eye. What's even more exciting, and this will get into what we're going to talk about in much of the rest of this talk, is how do you actually take several of these and put them together? How do you take the histology and the molecular, integrate that into a single understanding of a tumor, and then help to integrate that information to give it back to the clinician to treat that individual or specific patient? And I'm just gonna, this is a little advertisement for something that I don't make any money on it. It's free. We created an app that's on an iPhone that you can take a picture through your iPhone and run this algorithm and tell if the patient has a what kind of brain tumor it is, and if it has this mutation. And what we really targeted this at is underserved areas. What if they don't have sophisticated pathology or specialty pathology like neuropathology? This is an app that could just do this in under-resourced areas. And that's a direction that I really want to be thinking about moving. Well, everything I've told you about is related to cancer. But in fact, what I'm telling you about has just as much impact in non-cancer fields. So this is work that was done by a couple of my colleagues in Boston, the three on the right. And the woman holding her beautiful little child there had a miscarriage. The child was malformed and very abnormal, and she was very nervous about what was going to happen with the next one. And we were able to go in and sequence the mutation, or identify the mutations in the first fetus that did not survive, and we could then look into the genes of the baby she was carrying and determine that that baby was okay even before we could look at it with an ultrasound at a very, very early age. So this is using something called circulating fetal DNA, so it's in the mom's blood. You can take it out even early in the pregnancy, and we can start looking at this, and she was able to carry and very happy had this beautiful little baby. Now, this is all kind of cool, right? Kind of exciting, but what are the challenges to getting to using this in the clinic? And they are formidable. It turns out that clinicians are overwhelmed with data. As a matter of fact, there's a nice study that was done by a group called ARUP. It's a reference laboratory that showed that a clinician can use 5 data points at any time. Now, I was talking to a group of people earlier just today and saying, you know, when I go to the doctor and just have my annual physical, they get what's called a CBC, a complete blood count. They get a metabolic panel, that they do a lipid panel. They end up with about 30 different numbers, and they can use 5 of them. The rest of those are never used at all. How do we help them learn to use that data, or help them use that data more effectively? As I've already told you, there are millions of tests. In that our One Health system over 5 years, we had 500 million test results. Huge numbers that come in every day. And some of these are common, but it's easy to miss abnormal numbers or not know what to do with the abnormal numbers because you don't use those on a regular basis. More importantly, though, is that as testing is becoming more complex, so for many patients now, we're doing an entire genome. A genome is 3 billion base pairs. Each one of us has 3 billion base pairs in each of our cells. And we sequence those 3 billion base pairs. And we have to figure out, what does that all mean? And there are easily 2 to 3 million variations in any one of us. And how do we sort those and determine which ones are pathogenic and which ones are benign? It's a very complex thing to do, and certainly clinicians do not have the time to do this and sit down and go through those genomes. We have to help them do that. Currently, there's thousands of tests that they have that they can order. And again, most physicians don't even know which is the right test to order. We have to help them figure out, you know, if you have a family that has breast cancers, and you worry about a breast cancer gene like BRCA1 or BRCA2, there's a bunch of different tests to do the BRCA testing. Which is the right one for any one family? It's not always easy for a physician to figure that out, particularly if they don't do this on a regular basis. So how do we help them? How do we solve this dilemma? And that's really what I want to go through in the rest of this talk. And I'm going to start with an analogy. And my analogy is to maps. So for those of you who like reading fantasy, this is Middle-earth. And you can see the Shire up here. And this is Mordor, where they need to take, Frodor needs to take that ring to destroy it in Mount Doom, which is right here. And this map is great, right? It tells you where to go, where one place is and where another is. It shows you where the hills are. And so if you wanted to come through the valley and not go through those hills, you could do it. But it doesn't really help in terms of where the orcs are, or where other dangers might be, or what the best way might be given, you know, traffic jams. And so let me kind of advance to think about another map, and this is Google Maps. Now, Google Maps, as you can see here in LA, is great, right? Because it gives you a map of LA. And you can even look at traffic patterns. As you probably recognize, since this is all green, I had to get up at 2 o'clock in the morning to take this picture for Los Angeles. But it will tell you if I want to drive from Cedars-Sinai, where I started, to Cal State University, Dominguez Hills. It'll give me the best route at any given time to avoid traffic and to get here the fastest way. And I can even take that a little further. I can actually say, oh, I want to make a turn here, and here's what it looks like. So I even knew exactly what it was going to look like when I had to turn into parking lot 3 to park. So I was all prepared to get here. Now, the value of Google is that you type all of that into a single search engine. And what Google really created was not a database that had all of that information in it. Every one of those steps that I took you through, the map, the traffic, the directions, the Google Earth, is in a different database. The value, what Google brought to this was that they made all of these databases relational to each other. In other words, if you want to know where Cal State University Dominguez Hills is and what the elevation is and anything about the maps of it and an address to a specific parking lot 3 and where the boundaries of any of it were, all of these features, they're each in a separate database. They're different data types even, but they're relational to each other. We can actually start comparing them and using them to help us build the things that we want when we ask it, when we do a query in the Google search engine. Well, that's the concept of what I'm thinking about for healthcare. So we're going to take all of the data, even though it's orthogonal data types, image data, sequence data, clinical phenotyping data, what your physician writes down about you, where we can extract that and structure it using natural language processing. We can take all of that and put each of these in a different layer of data, but make them relational to each other so that we can then vertically interrogate that and then be able to really gain some information in complex ways about patients. So how does that work? So here's what we traditionally do. You know, historically, a physician will see a patient. So there's the clinical information. He gets the blood pressure and listens to your lung and your heart and all that stuff. And then he may order some laboratory tests. He may order some imaging data, an X-ray. He may get all of this different stuff. And he has to kind of, or she, I should say, needs to collect that data for themselves, think about how to best provide care to that individual, and then deliver that care. That's what we still do in most places. What we want to do, and actually, you can think about even simple algorithms that are used today. So, for example, if you want to, I heard about it today, if you want to ask about a glomerular filtration rate, that's not something you put in an analyzer and it comes out as GFR. No, it's a calculation. It's based on the amount of urine you make. It's a glomerular filtration rate is a measure of your kidney function. So it's the amount of urine you make and what kinds of molecules are in that urine. And that gives you, that's a simple calculations that's done. And so we can give that provider, the laboratory can give that provider information that goes beyond just measuring something. Well, if we take that a little farther, we can talk about some more complex algorithms. And that would be, for example, the sequencing of that tumor I showed you, where we had that EGFR mutation. Well, we sequenced that tumor, but we used a lot of information that came from external databases, from the literature, to know that that particular mutation actually was one that could be treated with that particular drug. And so when we did that, we created a more complex algorithm that we could provide back to that caregiver so that that caregiver could deliver better care. And as we take that even further, what our real goal is to collect all of that information. So take the clinical phenotyping, what the physician sees, integrate that with the imaging data, integrate that with the pathology data, integrate that with all the literature, and be able to then put together something that we can deliver back to a clinician that is going to help them better care for that patient, and even entire populations of patients. So let me give you an example of what I'm talking about. So this was work done from someone who was in my program in Boston. And this is that CBC, that hematology panel that has all these different parameters in it. And as I said, most physicians actually only look at one or two of these. For example, for the red blood cells, they might look at the hemoglobin here and the hematocrit just above it. Those are the two most common things that people look at. And they tend not to look at the rest of this. But what we did is we took a population of patients all with iron deficiency anemia. Okay, so now we're getting into population management. We took 10,000 CBCs on 2,000 patients, so an average of 5 results on each of those patients. And we asked an expert, a hematologist, someone who treats anemias all the time, said, what can you tell us, there was actually 2 of them, what can you tell us about these 10,000 CBCs on patients that have iron deficiency anemia? We even told them what the diagnosis was. All they can tell you is if that patient was anemic or not, and possibly that it was an iron deficiency anemia, but not always. We then trended all that data. So we put it onto these trends here. And when we trended the data, we said, now what can you tell us about all these patients? 2,000 patients. They could tell you when that patient was anemic and when they weren't. Okay? Everyone in this audience could do the exact same thing. What we then did was we went to the literature to understand the shape of the red blood cell. It's got this kind of biconcave shape, and how each of these different parameters, for example, the MCV, the mean corpuscular volume, something that most people don't even look at, the mean corpuscular hemoglobin content, MCHC. We looked at how each of those in mathematical models would change the shape of the red blood cell and how that would predict the average lifespan of that red blood cell. And when we did that, we could predict with 96% sensitivity, 88% specificity, the individual patient in that population of 2,000, who was going to be anemic 60 days from the time that blood was drawn. So all of a sudden, 2 months before that patient becomes anemic, we can tell you who in that population is going to become anemic, and we can ask that physician to say, you need to take care of this patient. It was about 15% out of the 2,000. So what does this do? Well, first of all, it allows you to focus the clinician's time. They don't have to see 2,000 patients. They only have to see about 200 patients, 300 patients. And so they have much more time to spend with each individual patient. They tell the rest of those thousands or 1,700 patients, you know what, you don't have to come in. We'll check your blood again, and you're going to be fine. And you don't have to take that iron that's going to cause abdominal cramps and constipation and all kinds of secondary effects. And for the patients that are going to become anemic, we can give you iron or EPO or a blood transfusion and prevent you from becoming anemic, prevent the complications of anemia, prevent you from going to the hospital. Remember, hospitals are bad places. People get sick in hospitals. And we can keep you healthy and at work. This completely changes the paradigm. By using that data more efficiently and more effectively, combined with mathematical modeling, we started managing these patients in a completely different way that was more cost-effective and gave the physicians more time with each patient to think through, why is this person needing to be treated whereas someone else doesn't? I'll give you one more very quick example. I'm not going to go through this in the same level of detail, but we did the same thing with diabetes. So now with diabetes, we follow something called A1C. It's a marker of a sugar molecule attaching to hemoglobin, and then it stays there. And if you have high sugar, high diabetes in your blood, you have more of this hemoglobin A1C. And if your hemoglobin A1C is 15 or 18 and you're diabetic, you're in trouble. You're headed towards end organ damage, blindness, kidney disease, peripheral neuropathies, all of these things. If your A1C is 2, you're doing great. But if your A1C is 8, you're kind of in the middle. And clinicians don't always know what to do. And it turns out we can tell them, because there's a population of people with an A1C of 8 that actually are under great control, and there's a population that are way out of control. And we can separate those out. It all has to do with the life. We figured out it's the life of the red blood cell. Everyone, we're taught in medical school, the average life of the red blood cell is 120 days. It turns out that it's variable. In some people, it's 50 days, and in other people, it's 150 days. If you're a 50-day red blood cell and you turn it over very fast, your A1C is going to look artificially low. And so you could have your real corrected hemoglobin, which is what is being shown here, could be that 15 if you had it at 8. But if you have a slow turnover of your red blood cells, it's the opposite. You may be under great control. It's just your blood stays around a long time, and so you accumulate more and more of that acetylated hemoglobin. So this allows us to really think about and deliver healthcare, not just to individuals, but think about how we manage populations of patients more efficiently and more effectively. And so this really takes us to the next stage, where it's not only integrating all of this information about the patient, but it's also about every time they come in, we get more and more data. And as we accumulate more data, our algorithms become better and better. It's because I like to say that machines and humans are opposites of each other. They work together great, but a human, I can read about a certain type of tumor one time, and I may not see it for 10 years, and all of a sudden it comes across my desk or my microscope, and I say, oh wow, this is that tumor I read about. In contrast, a computer's horrible at doing that. But if you take 1 million or 10 million data points and give it to a computer, it can figure out a signal in all the noise that's there. A human is horrible at that. That's what we're really bad at. So what we want to do is increase the amount of data, all the information we give to these computers, to basically work with humans together to be able to do the best job of taking care of patients. Oops, I already showed you. So how does this work in a healthcare system? So the input is all of our clinical information, pharmacy data, what kind of drugs patients are taking, illicit or not, laboratory data, imaging data, clinical phenotyping, but also external databases like the literature, genomic information from patients around the world, so that we can compare it to what is important for our patient, what's not. All of that needs to come in, and whether it gets stored in a cloud or a cluster or any kind of a system, it doesn't matter. I mean, they get structured differently, but we store the data. But now we have the data. And that data, just like I showed you early on with all those pretty little fluorescent dots, is worthless to a clinician. What we have to do is take that data and convert it into information using informatics. This is the first step where a clinical informaticist gets involved in taking that data and synthesizing it to understand it, to make it information. And then we have to, and remember that's just the sequence, to get to the specific gene and the mutations, the knowledge, we then have to go through modeling, inference, and prediction, basically applying our artificial intelligence and other types of mathematical algorithms to getting that knowledge that can then be used by our customers. And to get it to our customers, we have to make it actionable using things like decision support and integrated reporting, things that we have in our electronic medical record. And then physicians, laboratories, patients, increasingly patients want to be able to see and use their data. We want to be able to provide it to them, and to healthcare systems. How do we manage populations of patients, making sure we get the right patients to the right physician at the right time for the right therapy? That's exactly what we want to be doing to make our system more efficient. I'm not going to go through this in any detail. This is how we built it and structured it. Here's basically your laboratory system and your patient electronic medical record. You need a hardware and software. You need informatics. You need biostatistics. And then you need these knowledge pipelines to get that back into the hands of the physicians that need to use it. So just coming to an end here, and then I want to leave time for some question. What needs to be done? We certainly need to integrate and leverage the health and administrative systems. We don't do that right now, but we're learning to do that. This is an area of tremendous work right now and opportunity, I would say. We need to develop methods to integrate new and orthogonal data types. I've spoken about this several times, the idea of taking different types of data, whether it's image data in a TIFF file or a Word document file or a Excel spreadsheet with just numeric data. We need to be able to make those relational to each other so that we can use them with each other. And then we need to close that loop, basically make this information accessible to busy clinicians who are never going to be able to go through and look at the details that a clinical informaticist or someone else is going to be able to do. If we do this, what can we do? Well, we certainly can provide higher quality. Okay, we'll get diagnoses faster, we'll get prognosis better, we'll treatment stratify better. So we'll get higher quality, safer, and definitely more cost efficient. We'll reduce the number of days patients have to be in the hospital, the number of emergency room visits, the amount of time that people spend in healthcare, getting healthcare services delivered. This will, I think, support new ways of delivering healthcare. That's what I've believed all along, and I've talked about that a little bit. And it will support other aspects of the healthcare mission. And there I'm really talking about population management and really thinking about, different ethnic groups have different genetic variants. We don't want to misinterpret those. We need to find ways to really support the healthcare mission we have here. And I'll end by saying the need is clear. This is a huge opportunity. As you can see, this space of healthcare informatics has been growing at a very rapid rate, and it is over just the next couple of years, it's expected to add at least 50,000 new jobs in the United States. So this is an area where I think the opportunity to play a role in delivering healthcare is absolutely tremendous. So I will end there. I've left a few minutes. Thank you.
Please raise your hand, stand up, and ask your question if you do have any. Please, it's ours.
I'll break the ice.
Please, thank you.
Most of the students here are in laboratory science as their basic foundation. For this new field, what do you think is the type of course of study or what type of individual would be a good candidate to enter the informatics?
Yeah. So great question. If, in case everyone didn't hear it, the question is, what's the best field of study or the type of candidate that would be best for this field of clinical informatics? And the way I would answer, we've talked about this a little bit today with a number of people I've been speaking with, is that there is no one area or field or direction that you should be in studying this. Because for me, this is going to be a team science. It's going to require people who have expertise in coding and in informatics and computer science. But equally, it's going to require people who have an understanding of biology. And it's going to be people who have an understanding of patients and healthcare and diversity. All of these are going to have to come together as a team that's going to really make this work. And as I've been working with individuals here to think about a program between Cedars-Sinai, where I now work, and Cal State University Dominguez Hills, that is my goal, is to bring those kinds of teams of people together that each bring their own specific expertise and their own perspective to helping us solve these really complex problems. So it's not a simple answer. I don't think there is one way to do it. I think we need to look at many different avenues to bring that different expertise to making this work. Yeah.
Excellent presentation, really cool discoveries. What have you found to be some of the biggest challenges of getting your discoveries actually in use? Would it be the different types of electronic medical record systems, the different types of organization? You know, how do you get what you find into actually getting used by clinicians where you want that to happen?
Yeah. So the question is basically, how do you make all this actionable? How do you take this discovery and make sure it's being applied everywhere? And so there's a number of challenges in this respect. And you've mentioned some of them. The first one is that all healthcare systems are not the same. And it's worse than that. The most common healthcare system in the United States today is Epic. And the problem with Epic is that every installation of Epic is different. So when Cedars-Sinai, and I don't know how exactly they did it, but when they decided to do it, they, for example, put the patient's first name first and then their last name last. But when we did it back in Boston, we did the last name first and then the first name last. And so when you try and take an algorithm you build and deploy it from one system to another, you actually have to put these patches in to be able to reverse those or actually be able to understand what to get. So a major challenge here is that in the United States, we do not have a common or agreed upon structure of our electronic medical record. And a lot of other countries don't have this problem. We do. We have it in spades because it's not just one system. There's other systems, Meditech and others that people use, and ours don't talk to each other, and they don't talk effectively to each other. So that's the first problem. The second problem is the physicians themselves. So let me tell you an anecdote of something I did early on in my career and how it quickly changed physicians' behavior and why I'm encouraged that this is a solvable problem. This was back when I was at the Children's Hospital of Philadelphia, so this goes back like 15 years ago. And we were bothered by the fact that every child that was a year old or younger that came into the emergency room with a fever of unknown origin, so this is actually a pretty common thing to have happen, every one of them was treated according to the physician that was on call. Different protocols, different outcomes, very frustrating. So we got together and we came up with a single protocol. We said, if your child's less than a year old and they have a fever up to a certain number and they have no other symptoms, the only thing you can do are these 3 tests, and you have to do those 3 tests, and if those are negative, you send the patient home. And the physicians went crazy. They're like, you're taking away our ability to, practice the art of medicine. And it was horrible, and you can't tell us what to do. We said, well, we're going to tell you what to do. And after about 6 months, we said, okay, we've tried it, we're gonna get rid of it. And they're like, no, we have to do it this way. And what happened was that over that 6 months, the clinicians became more efficient in what they did. Any patient that had the kind of symptoms that fit this one very, very specific set of criteria got this testing. They didn't have any problems with it. If the tests were positive, they got admitted. If the tests were negative, they went home. It was very straightforward and easy. It reduced the amount of time they had to spend with those patients by about 68%. Not about, it was 68%. What it also meant, though, was that all the other patients that had something that was slightly different, the temperature was a little higher, they had a little bit of an eye drop or something like that, they had more time to spend with those cases that didn't fit the exact model. And they felt they were better able to care for those patients and take the time to recognize those variations in symptoms. So it will take education of the physicians. It's going to take the engagement of government to think about how to unify our healthcare records. And there's one more thing, is that we have to find ways that the FDA is going to find simple ways to approve of studied algorithms. Because right now they're not doing that. And you know, the FDA does a lot of really wonderful things, and they help keep us safe, but they do things that hurt us too. And the best example of that is COVID. For those of you who don't remember, and I remember because I was very involved in this, when our hands were tied to develop tests for COVID, the FDA would not allow it. They said the only test that can be done is the CDC test. It got distributed to 5 sites around the country, and it turned out there was a defective agent in their test. It was one of the buffers. And the test failed. And on March 2nd at 3 p.m., I still remember the date and where I was, the FDA said you can develop what's called an LDT, a lab-developed test, to develop your own test for SARS-CoV-2. And within a week, actually it was on a Monday, by Friday we had our test developed, and we actually had it deployed by Monday. So in a week's time, we were testing hundreds and eventually thousands of patients a day. We could have been doing that much earlier. The FDA did not allow us to do it. So we have to think about, how do we work with our government agencies? How do we work with our physicians? And how do we come up with a collectively agreed upon structure of our data in our electronic medical records? So a little bit of a long answer, but hopefully I addressed some of that concern.
So when it comes to patient confidentiality with this data, is there a higher risk of it being leaked? With switching conditions stuff?
Yeah. So the question is around basically HIPAA requirements and around patient confidentiality. And this is a very tough area. What I would say, first of all, is that the healthcare data has been, there's been massive leaks from different places, and that is a problem. But there's been massive leaks for financial data, like credit card companies and things like that. And so we have to find ways, and I think we're getting better at securing all the healthcare data. HIPAA does not protect against any data that's being used for clinical care. So if this is being used for clinical care, it's actually appropriate use and sharing of that data. So we can do that as long as we're doing clinical care. Where it becomes a little, for me, the most complicated is that if you put sequence, like if you sequence yourself and someone else gets that sequence data, they can identify you. There was a really interesting study that was published in Science probably 6, 7, 8 years ago, I don't know, maybe more. They showed that they took 6 random sequence data and they were able to identify that individual just based on the sequence. So there's a tremendous amount of information out there that allows you to do that. So you can identify people and what their risks might be. There is federal law that protects sequence data so that you cannot be in any way, it cannot be used against you. But I think that continues to be, that's the risk I see, is that this data cannot be de-identified at some level. Now, I'm actually a believer in openness for that data, and I shared with some other people a number of years ago, about 8 years ago, 5 of us at Harvard Medical School actually had our whole genome sequenced, and we put it out on the web, and I was one of those 5, and I'm fine with anybody knowing anything about me. My genome's actually pretty boring. Hopefully I'm not as boring as my genome is. We do have time for another question.
Yeah, because we have clinical laboratory scientist students here, what would you recommend to them to support the need for this bioanalysis that's needed now and more so in the future? How can they enhance their education, or what could you recommend for them?
So the question is how, for clinical laboratory science students, what's the need here from what I've talked about? And I would say it falls in 2 different categories. One is that I think there's going to be individuals that are going to want to go into laboratory sciences that are going to be informaticists, clinical laboratory informaticists. And that is a huge field. We're looking for people all the time that have that skill set to do a variety of different things in the laboratory. And so you become familiar with, laboratory sciences, how you, can work with a Roche Cobas instrument, whatever it is. But you also learn the informatics, how to hook that up to our EMR, how to actually manage that data, structure the data, use the data. But for the greater majority of people that go into these kinds of sciences, I think the key is to become familiar with it. Not that you're going to become an expert. I don't expect, and I say this all the time, including to residents that I had that went through our pathology training program, I don't expect you to code in Python. But what I do expect is every single one of you is going to understand where the data is, how it's structured, and how you access it so that you can use it when needed. And so I would encourage everybody to become familiar with how the data that you collect is collected, stored, cleaned, because it's often dirty, cleaned, and then how it is retrieved. And that's what I would focus on if I were going into the field today.
Question, Dr. please?
Hi.
Hi.
You mentioned earlier that, the quality of healthcare in the U.S. is much lower than other developed countries. And I'm wondering, how many of those countries do you think have adopted a more streamlined version of healthcare that's similar to what you're planning to do here?
So, question has to do with these other developed countries that are more effective in the use of healthcare, and how many of you have become more streamlined and adopted things in a better way than we have. Is that about right? Yeah, it's a complicated question because when people talk about this, it's not always easy to, you're not always comparing apples to apples. The number of MRIs that exist in Boston is the same number that exists in the entire country of Canada. So there's probably an exorbitant number in Boston, but there's probably not enough in Canada, and so people have to wait longer, and disease may progress farther, and so there's trade-offs of these things. Well, what I can tell you is that there is essentially one program in the country right now, that's Ohio State, the James Cancer Center, that has switched to doing digital pathology and applying algorithms onto it. In Europe, there's probably 100. So they have gone and adopted digital methodologies and algorithm applications in Spain, in France, the first one was in the Netherlands, in Belgium, in Israel. I mean, and I've visited a number of these. They're all way ahead of us in terms of doing things more efficiently and effectively. So there has been a contribution. I don't know exactly what it is in terms of, quantitating that.
We have time for a couple more questions, but we have to stop. So please stand up and speak up and ask your question.
Hi. So when you were talking about these slides that you would be putting on, like the specimens you would put on a slide, and then you'd go through diagnosing the patient, but also how were you able to know like what was going to happen in the future, or even the example with the iron deficiency? How using it, especially using an app, how are you able to tell like what's going to happen not just now or a week from now, but a really long time from now?
Yeah. So the question is, how do we know, how can we predict the future, right? How do we have a crystal ball? And it's different for different things. So for example, the anemia model that I talked about was based on a combination of different data from biology, understanding the red blood cell, the shape of the red blood cell, and how changes in the shape of that red blood cell would affect the life of the red blood cell, how that life of the red blood cell would then affect the amount of blood that you have, the amount of red blood cells you have in your body. And what we then did is layered on top of that the patient's individual blood counts and how each of those different parameters would change, either decrease or not change the life of that red blood cell. And if it was going to be decreasing the life of the red blood cell, we could predict that that person would become anemic. And those predictions, based on looking at 10,000 results, were actually extremely accurate. So that's how we could make that prediction. Now, with regard to looking at a glass slide and predicting what is going to happen to the patient, we have years and decades of historic information that if you have a grade 2 glioma, a type of a brain tumor, this is the course that you're most likely to follow. This is how long until it's going to repeat symptoms or recur. This is how long you have to likely live, the average time you're going to live. And then so that's the first level of what we had. But what we were able to do is use the computer to actually tell us if there was a specific mutation. That the one is in IDH1. And if you have a mutation in IDH1, you actually have a better prognosis. You actually do better. So we knew then from that historical data that when we could not only say that it's a grade 2 glioma, but it's a grade 2 glioma with this particular mutation, we knew that that had a different trajectory, a different Kaplan-Meier curve of what's going to happen to that patient over time. And so that gave us the ability to look into the future, and those have also come out to be accurate. So it's a tough question, but
I'd like to ask a last question, and I don't have first-hand information about this, but I know that the students will come to me and ask me, so I would like to ask you. A student with bachelor's degree, let's say with 1 year of training in bioinformatics, could you please tell us what is the salary range?
You're not going to answer that? Yeah.
So
So bioinformaticians that are coming out of with a bachelor's degree, and particularly if you have a little bit of training, so like let's say an extra year, starting salaries are somewhere around $75,000 to $80,000 and can go up to $100,000. That includes all your, with that comes benefits and everything else, which is generally about 25 to 30% of that salary. So salaries are starting, if you include everything, somewhere between $100,000 and $130,000 a year. But the actual salary is probably 75 to 80 at the low end, up to about 100,000 for your starting salary.
I'll take your names whenever you're ready. Please join me to thank Dr. Golden.